Weil and Brady, in their book The Nature and Properties of Soil, stated that if we were small enough to enter the tiny passage of the soil, we will encounter a very diverse world populated by microscopic microorganisms. All those microorganisms that live in the soil are fiercely competing for every root, leaf, or plant debris that they use as a food source. In a handful of soil, we are likely containing billions of microscopic microorganisms. From those, bacteria is the most numerous. It's been estimated, for example, that in 1 square meter to a 15 centimeters depth in a fertile soil, 40 to 500 grams of biomass it will come from bacteria. Unfortunately, among those microscopic microorganisms living in the soil, not all of them are beneficial. There are some of them that they are harmful to plants or they are known as a plant pathogens. My name is Jose Garcia. I am a fourth-year PhD candidate working with Dr. Steven Redau in the School of Plant and Environmental Science at Virginia Tech, and today I would like to introduce you to anaerobic soil disinfestation. Anaerobic soil disinfestation is a technique independently developed in Japan and the Netherlands in the early 2000s to manage soil-borne plant pathogens. It consists by applying cardboard sources, for example, wheat bran, rice bran, moisture compost, among others, followed by irrigation to full capacity and covering with plastic tarp or any impermeable film that create anaerobic conditions. Now, these anaerobic conditions shift microbial community to facultative anaerobic or aerobic microbes that produce volatile compounds or organic compounds that they are toxic to most soil-borne plant pathogens. Currently in Virginia, besides the carbon sources already mentioned, we are currently researching in the use of cover crop biomass as alternative for carbon sources. And this will allow us to be more practical for a large-scale productions and to be less labor intense. In recent years, we have seen an increase of Sclerotium rolfsii throughout the Mid-Atlantic region. Sclerotium rolfsii causes southern blight in vegetables and other crops. This pathogen is characterized by producing a white fluffy mycelium, which produces these little round structures called sclerotia. Sclerotia can survive in the soil from months to years. The pathogen commonly attacks the plants at the base of the main stem at the soil line, subsequently killing the whole plant. As we can see in this bag, the pathogen shows the aggressiveness by growing without being there a lot of food source, and we can see the mycelium and the sclerotia being produced. One of our main objectives in our studies is to test whether this pathogen can be managed through the use of cover crops as a carbon sources in anaerobic soil disinfestation settings. Before to test anaerobic soil disinfestation in the field, we are performing a series of growth chamber experiments. The process for certain this experiment is summarized in this picture. In an empty container, fresh chopped cover crop was mixed in a soil collected from the field, subsequently covered with plastic mulch and incubated for 2 weeks in a 60 to 77 Fahrenheit temperature with 12 day and night light period. This was with the intention to mimic spring temperatures throughout the Mid-Atlantic. This table is listing the cover crops and the fresh weight rate used in the growth chamber experiment. At least one grass, brassica, and legume cover crop was used, and we also include some additional carbon sources, for example chicken poultry litter, biochar, and moisture compost as an option for organic productions. These pictures show an overall view of the tomato transplants in the greenhouse after the soil was treated with anaerobic soil disinfestation. As for some preliminary results, these pictures show how the tomato plants are looking 7 weeks after being transplanted in the greenhouse post anaerobic soil disinfestation treatment. This is a dwarf tomato cultivar, therefore we did not expect to see much growth of it, but we can determine whether there is a negative effect due to the carbon sources or the anaerobic soil disinfestation treatment. The good news is that no phytotoxicity or other negative effects were observed. However, we can visualize some difference in the leaf colors or tomato fruit serine that are due to the carbon sources effect compared to the anaerobic control. By looking more specifically at the preliminary result, this bar graph shows the effect of the carbon sources and the anaerobic soil disinfestation in the tomato shoot biomass fresh weight. As we can see by the statistical analysis, there is no significantly different among carbon sources. However, numerically, we can see that chicken poultry litter and mushroom compost were the two treatments that numerically have the greater shoot biomass. On the contrary, rapeseed, Austrian winter pea, and biochar, together with one of the control, have the less shoot biomass. By looking at the effect of the carbon sources in the tomato fruit, this bar graph showed that carbon sources did significantly affect the tomato fruit weight. When wheat was applied as a carbon source, we have significantly greater fruit production compared to the rest of the carbon sources. On the other hand, biochar and brassica cover crop have the least fruit yield together with the aerobic control. And lastly, because the soil was mixed with inoculated millet seed infested with Sclerotium rolfsii prior to anaerobic soil disinfestation application, we wanted to test whether there is an effect on southern blight. And interestingly, there was no significantly difference among treatments, including the control, by looking at the pathogen severity. The severity was evaluated in a scale from 1 to 5, where 1 is a healthy plant and 5 is a completely dead plant. For this particular experiment, numerically, sun cover crop produced greater disease severity than where there was no carbon source applied, and that hairy vetch, mushroom compost, followed by two grasses cover crops were the treatments that had less disease severity than the controls. But this is only numerically, not statistically significant. We are still currently working on analyzing other variables measured, for example, properties on soil, pH, nitrogen content, as well as performing another rep of the experiment to fully understand the surprising of some results. But with these preliminary results, we can begin to understand what could be the potential option of sun cover crops of other carbon sources and aerobic soil disinfestation settings in Virginia and Mid-Atlantic region. With this, I would like to thank to my major advisor, Dr. Steven Rideout, and my committee members, as well as the organization that are supporting and funding these studies. If there is any questions or additional information needed, please feel free to reach us to the Eastern Shore Area website.